Thank you, Vijay. What I plan to do within the next hour and a half, with luck less than that, is to take you very rapidly through a view of evolution, which I think is shared by most biologists, certainly by most evolutionary biologists, but I can't promise that that's the case. So it's going to be a pretty personal view. And for the experts in the audience, I apologize at the outset because this is going to be a very superficial view as well. And the constraints of time make it impossible for me to put in everything that I would have liked to put in or that experts would have liked to hear. So please bear with me on that account. This is meant only for people who want a quick rush through the ideas that drive evolutionary biology today. And I have framed my talk along the following lines. First of all, what is it that we seek to explain when we study evolution? Broadly, the existence and functioning of what you might call living matter, that is to say us plus the plants and trees and microbes and bacteria and frogs and fishes and so forth, which are all extremely improbable states of matter. I won't expand on this point any further, but I will take it for granted that this is something all of us would agree on. Now, when you think of it in this way, and especially when you look upon living matter from a physical scientist's point of view, the default option for explanation, of course, should be physics and chemistry. However, it turns out that the actual default option which is used by evolutionary biologists is something quite different, which is the so-called principle of evolution by natural selection. Therefore, in this talk, I will build on ideas underpinning natural selection first, because that's the way people, evolutionary biologists, look at evolution. And then I'll come back and spend some time on these other ways of looking at evolution, which are now called alternatives to natural selection. In fact, though in fact it should be the other way around. These other options should be the first option and natural selection should have been the alternative. But this is the way the situation is. So that's living matter. All kinds of colors, shapes, sizes, behaviors, things that they eat, creatures that eat them, you name it. And these are classified in a variety of ways which are really somewhat staggering when you look at the detail to which such classification has gone. And the first observation that one makes when one looks at living matter, when one looks at the living world, is that one can classify them into nested categories, which means you have a group, then you have a larger group which includes many of the smaller groups of the first kind, and then you have a yet larger group which includes many of the smaller groups of the second kind, and so on and so on and so forth. Among these groups, the one that evolutionary biologists set most store by is the so-called species. Now, the definition of species is somewhat tricky, and I'm not going to go and attempt it, but somewhere during the course of this talk, we'll see a place where species come into being. Now, there are these larger groups, genus, order, family, kingdom, phylum, and these days you even have words like supergroup based on molecular studies. It's not very clear how objective these classifications are, but nonetheless, these are things that people work with. And by and large, these classifications, all these classifications are based on the same system as any classification, which is things that fall in a group are by and large more similar to each other than things that fall outside that group and therefore belong to a different group. So the first question you might want to ask is, how many living forms are there? And the numbers vary a lot depending on who you talk to. Some ecologists, in fact, estimate that number as 100 million or thereabouts. Others estimate this number at many million. And again, the method of counting and therefore the method of categorization varies from individual to individual. One thing that you can say for sure, however, is that the distribution of forms, irrespective of how you categorize them, the distribution of forms across these various categories is very non-uniform. And you see some patterns, some of them are visible there on the screen. So for example, among animal forms, the so-called arthropods are the overwhelming majority. And among these arthropods, insects are in the overwhelming majority. Among plant forms, flowering plants, angiosperms are in the overwhelming majority. So there are very clear skews visible in the distribution of living forms across these categories. And the numbers also, as I said, vary a lot from category to category. One further generalization that we can make is that if you classify forms crudely into single-celled forms and many-celled forms, and within the single-celled forms into bacteria, that is cells that lack a nucleus, and today one would say bacteria and archaea, and many-celled forms into also single-celled forms, beg your pardon, the forms with a nucleus also into single-cell formed and many-cell forms, you come up with the following generalization. The overwhelming majority of all the species that are here on Earth are bacteria and archaea, that is single-cell forms lacking a nucleus. Among what is left, the vast majority consists of single-cell forms. And then among what is left, you have these skewed patterns that I told you about. One quick statement now. So far, we have absolutely no theoretical understanding of why the number of forms, number of living forms, if you wish, the number of living species, or the number of phyla, or the number of genera, is what it is. We have no idea of why the distribution among these forms is as skewed as it is. And we also have no idea why bacteria, which are by many criteria among the most ancient forms of life on Earth, the bacteria and archaea, seem to predominate the landscape, as it were. So why is it that evolution has not gone further? And we can discuss possible reasons for this later as we come along. The second thing, yes please. Oh, I didn't go into detail. So this is based on certain models. You see, you cannot count every species there is. So you build models, you carry out techniques of sampling, and based on those models, you make certain predictions. So it's the same kind of thing that you do whenever you have to take any sample. And then you go back and ask someone else to do an independent count in certain cases to see how well it fits your model. That's all. That's what I said. So this depends on sampling techniques. I'm not going to go into these details here. Right. So when you look at forms, the second thing that you notice, and this was done, of course, many years ago, in fact, more than a couple of 100 years ago, close to 300 years ago, you see that within any given category, the forms can be ordered, and they can be ordered by imposing on them a metric which seems to climb out of the category itself. In other words, within a given category, there seem to be certain individuals that are more closely like each other than other individuals. And based on this closeness of relationship, people started building trees of the sort that you see here. This is a famous example from the history of evolutionary research, which is the evolutionary lineage of the horse, which is, as it happens, among the best studied lineages in evolution. So you can build patterns like this, and you notice already that in this pattern, without any theoretical underpinning, you see lines, and then you see branches, and then you see further lines and further branches, and so forth. The culmination of this kind of ordering and categorizing is what has come to be called the tree of life, which is an attempt to basically indicate every living form. Obviously, you can't do that. So every living species, you can't even do that. But type examples of living species, so you have broader and broader categories, you try and put them on this sort of a branching network of the same type that you saw in the previous slide. And the crux of this is that you can do so, that it's possible to fit organisms at various levels onto a kind of branching tree. And it became tempting, since the first people who looked at such trees saw that the trees seemed to proceed from simple forms to more complex forms, the way they had put it. So this was simply an outcome of classification and ordering. It became tempting to think that this tree might in some way indicate a transition of forms in ways that weren't clear from simple to complex. The second thing that people noticed immediately, and this followed the categorization itself, was that living forms seem to display what one calls adaptation, which is they seem to display the sorts of characteristics that an engineer would have endowed them with, an engineer who built these forms would have endowed them with, had he or she known what those forms were meant for. In other words, living forms seem to indicate features of design, even though no one could figure out where that design came from. So for example, beak shapes in birds, obvious products of design, because if you study these various birds, you see that some of them have beaks which can crack nuts, others have beaks that can peel barks, yet others have beaks which have the right length and curvature to go deep inside and reach the nectary of a flower and draw out nectar. Others have beaks that can work like pincers and pull out insects from deep inside trees, and so on and so forth. So this again is something very, very well studied, and there's no doubt that these are adaptations to picking up various kinds of food. Mimicry in butterflies, another famous example. So here you have all kinds of butterflies shown here on the screen, but I direct your attention to two classes, one on the top and one below. And broadly, it turns out that these two exhibit the following features. On the one hand, you have butterflies that are poisonous, and all these poisonous to birds, that is, the animals that feed them most often, and all these poisonous butterflies seem to exhibit the same kind of body pattern. So there's some sort of convergence in body pattern. And the idea there seems to be that if you eat one of them, or if a bird tries to eat one of them, it feels the poison, feels distaste, and therefore avoids eating others of them. So there's mutual benefit. That's adaptation. The second kind of mimicry there is more interesting. It's the so-called Batesian mimicry, where you have a poisonous butterfly and a second butterfly which is non-poisonous, imitating the poisonous one in form and quite often in behavior as well. So if a bird knows that that particular body form is associated with something bad, associated with poison, it stops eating not only the poisonous form but also the non-poisonous form, which is in a way getting away with a dishonest signal. But the point that I want to make here is that this is an obvious adaptation. This is something that helps. So you can see immediately the moment you see it, aha, this pattern must be for self-preservation. This is a rather unusual example, but one that I like. This has to do with what are called circadian rhythms. On both the figures at the bottom, the x-axis is activity patterns. The black bars show periods of activity, and the empty spaces show periods of doing nothing, and the vertical line shows time. So basically what this is trying to tell you is that there are patterns of activity followed by patterns of rest in these ants that were being studied by Sharma, Chandrasekharan and others. This was work done right here, by the way, in Bangalore. And the interesting point is that as with a great many living forms, these patterns fall into near 24-hour cycles, even in the absence of a light-dark stimulation. In other words, even in the absence of a normal day and night. But Sharma and Chandrasekharan found something very interesting. They found, and you have to look at the slide a bit closely for that, compare the top half and the bottom half, the top bars and the bottom bars. They found that when they compared these rhythms in two so-called castes of ants, this is the common ground ant Camponotus, they found that the same sort of ant, something called a major worker, that functioned as a soldier was arrhythmic. That's the bottom thing where you see black and white interspersed all over the place. Whereas the same the same major worker who worked on the nest, taking care of the nest, had a 24-hour rhythm, which you find in that clear dark lines that are shown there. What's the point here? The point here seems to be that the soldiers need to be on guard all the time, whereas the workers who are at the nest need to feed the larvae or can get away with feeding the larvae only at 24-hour cycles. So the soldiers have become arrhythmic here. And once again, you see kind of sense leaping out of this data. So these explanations for all these varied observations for the tree of life itself, plus for this overwhelming signature of adaptation that living forms seem to present, these observations were responded to, one might say, by various explanations. A creator who created everything in one go, rational morphology, which is properties of physics and chemistry, roughly speaking, came next historically. Multiple creations were spoken about by others. This happened after fossils started appearing on the scene and people saw that everything couldn't have been made at the same time. Inner drive essentially refers to a notion propagated by Lamarck that animals, and less so plants, try to improve themselves in some way, and in the course of this improvement they change. And finally we come to natural selection, which is this theory originally propounded in something like its modern form by Darwin and Wallace, which has now been modified into something else called the modern synthesis, which basically includes ideas put forward by Darwin and Wallace, along with the very strong underpinnings that have come from genetics, biochemistry, physiology, ecology, paleontology, developmental biology, and most recently, of course, molecular biology. However, the modern synthesis, even though today it rests very much on molecular biology, on what we know about how genes and proteins function, even though it comes to rest very much on molecular biology, it turns out that its formal structure was more or less finished by the 1940s. And it's that formal structure that I'll proceed to examine in the next few slides. The essence of the modern synthesis, by the way, this is also called the evolutionary synthesis sometimes, also called neo-Darwinism sometimes, all of them mean the same thing. The essence of this modern synthesis is that the tree of life that we saw earlier is a tree of descent. It says that this closeness in form that you see between plants and animals is because of a closeness of relationship as measured through ancestor-descendant patterns. And it says that in the course of this descent from an ancestor to descendants, in the course of this pattern of heredity, forms changed. And Darwin's own capsule description of what natural selection was all about was descent with modification, with natural selection serving as the mechanism which explained this modification. And as the modern synthesis developed, Darwin of course had no idea of genes. As the modern synthesis developed, one began to see that genes played a very important role in defining forms, and therefore the modern synthesis, meaning the explanation of evolution by natural selection, came to be seen as a theory which accounted for the change in gene frequencies that accompanies changes in forms. Today, of course, we know a great deal about what genes and what genes are and what sorts of games they play. We know that there are DNA molecules organized into chromosomes. Genes are strings of DNA. DNA codes for proteins, and so on and so forth. Proteins build bodies, catalyze transformations, and so on. And underpinning all of molecular biology, there's something that Crick called the central dogma, which has to do with information transfer, which goes from DNA to RNA to proteins in the normal course of events, can also go from DNA to DNA, RNA to RNA, RNA to DNA. But broadly speaking, nucleic acid to nucleic acid or nucleic acid to protein, never in the reverse direction. And as far as we know, this remains valid, even though it has an unfortunate name. Right. Now, natural selection, this theory presented by Darwin and Wallace, essentially rests on the non-random differential survival or reproduction of individuals that differ from each other. The word phenotype simply refers to all these characteristics of individuals that you can spot. So the differences of interest to you are phenotypic differences. This differential reproduction, so again, this is the theory of natural selection, this differential reproduction is said to be based on heritable differences. In other words, to be based on differences that can be passed from parents to offspring. Because there is differential reproduction, the representation of these heritable traits changes. And because this representation changes, the genes that underpin these heritable traits also change. So you say indirectly you have an effect on genes, even though natural selection works directly on the phenotype. The simplest situation, which is the one that a lot of early natural selection theory concentrated on, involved a slowly varying environment and invariant mapping from genes to properties, genotype to phenotype, as it said. Additive effects, which meant that you could look at genes one by one. Fixed fitnesses, which means fixed values for these propensities to reproduce with a certain efficiency, given a certain genotype and in a certain environment. And very importantly, the idea was that in the course of this reproduction, what was actually going on, not intentionally of course, but what was actually going on was this sort of engineering fit to design, where the fit was to a set of requirements imposed by the environment of the organism, and the changes which were made ipso facto resulted in improved attempts within quotation marks, improved attempts by the organism to fulfill those requirements imposed by the environment. So, to put it in simple forms, if the environment in which the organism was involved moving through air, you had improvements in flying. If the environments involved moving through water, you had improvements in swimming, streamlining, for example, in both cases. If the environment involved moving on hard ground, you had improvements in the musculature necessary for walking or running or hopping or whatever the mode of movement was. So basically, the way one could look at evolution, was that the environment imposed certain requirements. These requirements were fulfilled by natural selection. The picture, the real picture, of course, is not that at all. The real picture involves random changes, I'll come to that in a moment, in the genetic makeup of organisms. And these random changes sometimes result in advantageous effects, sometimes result in disadvantageous effects. The advantageous effects are picked up, the disadvantageous effects are discarded, and therefore you keep moving up. And the metaphor which was used very often to describe this kind of process was hill climbing. And one of the most popular pictures in evolutionary biology is that of the so-called fitness landscape. So this is like a standard topographic map which all of us had to do as our school homework. The pluses there indicate places where the design of the organism to its environment is optimal, therefore places of maximum fitness standing for net reproductive efficiency. And below the pluses, of course, below these mountain peaks, if you wish, there are valleys. And if you move through a valley, you can reach a second mountain peak eventually, and so on and so forth. So the metaphorical picture here, one sec please, the metaphorical picture here was of species, organisms of a kind, distributed in a fitness landscape that was peculiar to them, trying, again within quotation marks, trying to reach at least the local peak, which natural selection told you immediately how you could do, or ideally the best peak in which you could be optimally fitted to that environment. Yes, please. That would need a lot of discussion. That has been looked at very carefully in many cases where it is known that it's true. All the attempts that have been made so far to look for directed mutations have failed, meaning that the results have been negative. The only situation in which I know you can get directed mutations are in the lab, where today we routinely engineer mutations to serve certain purposes.
Hello. The phenotypic variation of Darwin, that has got no connection with the genes. So how this phenotypic variation proposed by Darwin at that time, and how can we connect with that with the variation of the genes?
Right. Well, Darwin didn't know anything about genes. In fact, one of the ironies of the theory of natural selection is neither Darwin nor Wallace knew what heredity was based on, nor did they know how genotypes were converted to, what we would now call genotypes, were converted to phenotypes. Now, today what we know is that genes underpin phenotypes. To what extent we will see presently. Genes make proteins, or encode proteins if you want to be accurate, and it's the conglomerate of proteins and their associations that make up bodies. So this is the way genes relate to phenotypes. And the primary source of variation, which of course was a mystery to both Darwin and Wallace, even though speculated, the primary source of variation among those who accept the modern synthesis is believed to be random variations in genes, which result in variations in phenotypes, on which selection acts. Therefore, indirectly it acts on the genes. Right. Now, is there evidence for natural selection? This is the summary picture that I've presented. Huge numbers of evidence. This is a classical example from the evolutionary literature of something called industrial melanism. And if you look at the two pictures, you see a dark-looking butterfly on the left, a moth, a light-looking moth on the right-hand side. It turns out that the left also has a light moth in the same picture. The right also has a dark moth in the same picture. It's that the two are camouflaged. And the reason they are camouflaged respectively is that the left picture was taken at a time, as it happens it was taken very recently, but we'll try and pretend it was taken in the late 1700s, at a time when the wood of birch trees and other temperate trees in Western Europe was relatively light colored. The picture on the right-hand side was taken when the wood had turned dark thanks to the Industrial Revolution. So on this dark wood, the white form stands out. On the light wood, the dark form stands out. And in fact, there's a recorded evolutionary transition from the prevalence of the white form to the prevalence of the dark form. And today, as I said, thanks to anti-pollution measures, a return of the light form once again. And this has been tracked in the course of time, fortunately, because you've had a great many people observing, the amateur naturalists observing these butterflies. And fortunately, we also know the genetic basis of this transition. So this is a somewhat unusual example. These are, yeah. You have to look for it, yes. So look for it when you see the slide. So that shows you how well the camouflage is. Okay. Now, there have been a number of observations carried out over the last 30 or 40 years, and there's really been a dramatic improvement in our knowledge of evolution in action. Number of cases studied of natural selection actually going on. So what is it that people do? So they monitor the study population, look at it in terms of variations in respect of certain characteristics, come back a generation later, look at the distribution of those very variations, those very characters, come back 2 generations later, look at those very variations and so forth. And therefore, track these variations as a function of time. Now, with clever statistics and some belief in the fact that these variations are underpinned by genetic variations, and in most of these cases this belief is valid, you can actually carry out what's known as a regression analysis and figure out how much of this variation can be ascribed not due to chance events or environmental factors or something, but actually due to genetic changes. So this fraction of variation that can be so ascribed is called heritability. So you can compute heritability. From that you can compute the intensity of selection. Very brief lesson from all of these 4 pictures, which you can study in detail later, is that by and large, selection seems to work just as well for decreases in traits as increases in traits. If you look at the dashed lines in these 4 figures, and they refer to different traits, as you can see that by and large these figures are symmetrical. So they show the frequency distribution of various intensity of selection, and the intensities themselves are on the x-axis. So that's one lesson. The second lesson is that if you look at the continuous lines here, which are measurements of body size in the same experiment. So the experiment actually concentrated on one or the other aspect. Fecundity is something that you can look at there, for example, but also looked at body size. It turns out that body size, by and large, seems to be under directional selection in these cases. So bigger is better, as the saying goes, which has turned out to be a useful generalization in evolution. This is a famous example. I show it here simply because the person who did it was the father of Roger Penrose, who is known, I think, to many people in this institute. And this is an example on fitnesses, in human babies, measured in terms of early infant mortality. So once again, you get a frequency distribution as a function of weight in this case. And you see that the maximum fitness occurs in this case, not for the maximum weight, but for an intermediate weight. So here you have an example of what's called stabilizing selection. Babies that are underweight are at a risk of dying early. Babies that are overweight are also at a risk for dying, early. Therefore, the intermediate weight is the best. Another famous example, worked out in great detail in terms of its implications for genetics. If you look at the 2 maps, you see this striking correlation between malaria-prone areas, at least areas which were malaria-prone historically, and the prevalence of a particular form of genetic constitution in human beings known as sickle cell anemia. This sickle cell anemia carries with it a correlate called a sickle cell trait, where you have 1 copy of a normal gene and 1 copy of a mutant gene, a variant gene, which turns out, this combination turns out to confer protection against malaria. And you have the prevalence of this trait precisely in those regions that are malaria prone. And this pattern of overlap between where malaria is frequent and where the sickle cell gene is frequent was first discovered by a person called Allison, and then the evolutionary, quantitative evolutionary consequences were worked out by JBS Haldane, and continues to be an area of active study. So once again shows you that natural selection has been at work in order to keep this gene in place, because in those areas where there is no malaria, this gene is missing, this genetic form is missing. And the reason it's missing, of course, is because it makes you anemic. So it's really not good for you, except in those places where you have natural selection. So finally, This is the picture of evolution by natural selection. The green thing stands for the environment, the green color stands for the environment which permeates everything. So you have genotypes. These genotypes express phenotypes. So this is the job of physiology, biochemistry, developmental biology, you name it. Selection acts on these phenotypes. Certain phenotypes are more successful than others at survival or breeding or both. As a result of which, the representation of phenotypes in the next generation has changed, and therefore the representation of genotypes as well has changed. Behind all of this, you have variations in genotype, which, as I said, occur randomly. The bulk of these variations occur due to shuffling of genes, due to what's called recombination, but this does not introduce any novel genetic material. Some of these variations occur due to what are called mutations, which does introduce novel genetic material. And the role of the environment here, as I said, of course, is to sift, is to pick and choose between those that are successful and those that are less successful. Selection can work in two ways. It can be very stringent. So there are two of these people who are running away from the tiger. Either both can escape if they run faster than the tiger, or neither can escape if they cannot run fast enough. And this is called hard selection. On the other hand, there's something called soft selection, where the critical thing is to run faster than your friend or someone who was your friend. Not necessarily faster than the tiger, because the tiger can catch just one of you. Both of these things can work. Their implications are rather different. With hard selection, the environmental requirements are stringent, so everyone may make it or everyone may not make it. And among those who make it, of course, you may get competition later. With soft selection, only the best of the lot get through, irrespective of how good they are in an objective sense. Now, the idea of natural selection has various embellishments put on it. Darwin himself put on an embellishment called sexual selection, which I will not go into here at all, except to say that Darwin used it to explain why in a great many species A, males differ very much from females. So this environment, which should really be neutral, which should be agnostic as far as individuals are concerned, seems to make a difference between males and females. And secondly, males seem to exhibit what Darwin called exaggerated traits, like huge sizes, elaborate antlers, the famous example of the peacock's tail and so forth, which seems to exceed anything that a reasonable theory of natural selection would account for. And both of these Darwin explained by saying that on the one hand, there's an asymmetry between males and females, and the asymmetry lies in the fact that females devote more energy per act of reproduction than males do, per unit reproduction. And the simple way of seeing what this is to look at the relative sizes of the egg and the sperm, let's say in mammals. This is about a factor of easily 1,000. If you go to things like frogs, it's a factor of 1,000,000. So females invest 1,000,000-fold, or in our case, about 1,000-fold as much as men in any act of reproduction. Therefore, this is asymmetric. Because of this asymmetric, males compete amongst themselves for access to females. So there's a nice kind of reasoning which goes behind this, and again there's a rich area of study, but I won't go into this. Basically point out that males and females differ. J.B.S. Haldane pointed out that natural selection could also act indirectly, meaning not act on an individual, but act on an individual's relatives if that relative happened to share genes with this individual, which of course relatives generally do, at least close relatives usually do. And he expressed this in the form of this saying, which is quoted all the time, saying that he would be willing to jump into the river at the risk of losing his life if the life he saved was that of his brother. But then his jumping, you could bet on his jumping to an extent of 50%. If the life he saved was a first cousin, then the chance of his jumping would be 1/8, and so on and so forth. In other words, the probability of his jumping to give up his life would be in proportional to this relationship. I made a small mistake there. So jump into a river to save his brother if he could save at least 2 brothers. Jump in to save his cousin if he could save at least 8 cousins, and so forth. 2 times half being 1, 8 times 1/8 being 1, and so on and so forth. Good. Yet another modification which came on fairly early was due to a man called Sewell Wright and was picked up by others. And I go back to this fitness landscape picture once again. And here the idea is, if the fitness landscape has multiple peaks, and if you are convinced, based on your look at things from an engineering perspective, that organisms have reached, a population has reached the highest peak, how does it do so, given that it can get trapped at intermediate peaks? And Wright said, look, it may get trapped at an intermediate peak, but then demography might come to your help. And this was a new element in the theory of evolution by natural selection. Demography can come to your help because if you have small population sizes, this random assortment of genes that we call recombination can sometimes throw up chance combinations, right, that enable you to cross valleys and ascend neighboring peaks. So basically chance coming to your aid. Again, I won't go into details here. We'll look at the consequences of chance in a different, from a different perspective slightly later. So this way what you can get is adaptations, maladaptive combinations due to chance, but these maladaptive combinations, because they go down the fitness valley, enable these populations to succeed and climb up some other fitness peak. And he called this a shifting balance simply because you have one balance, one sort of equilibrium spectrum, if you wish, of phenotypes and genotypes, giving way to a second balance, so the balance shifts gradually, and eventually he said the ultimate, the highest peak would come to be reached. Right. Huge amount of mathematics went into the modern synthesis and continues for that matter to go into evolutionary biology, evolutionary theory, which I will not get into right now. And except to point out that the kinds of mathematics that went in were rather different. Population genetics, which looked at gene frequencies, which by and large made use of algebra, difference equations, calculus, differential and integral equations came foremost. So it looked at single genes, the effects of single genes, the effects of combinations of genes, and interactions between genes. Then there's an entirely different school of mathematical approaches to evolutionary biology called quantitative genetics, and in fact it precedes Darwin. That's somewhat interesting. Precedes the theory of natural selection, which deals with statistical concepts like variance, heritability, regression measures, and so on, where the gene itself is lost. It's nowhere in the picture, but the average gene pool in one population at one generation is compared with the average gene pool, or I should say with the consequences of the average gene pool in the same population in succeeding generations. And I gave you an example of a regression approach. These days, there's a huge amount of mathematics being used from a statistical mechanics perspective, where the kind of approach that people have been tending to use is to say, here are all the possibilities open to these organisms. Is there any way in which I can statistically estimate the optimal combination of these opportunities that would fit the situation? Fisher sort of categorized these different schools rather nicely. He said you could explain evolution in terms of known genetical causes, so make certain assumptions as to the properties of the underlying causes and then work out their consequences. Or, and he said he preferred to do this himself, explain the genetical phenomena themselves on the basis of evolutionary principles. And there's a rather nice distinction between these two approaches. Right. The final big question, of course, which we have not addressed at all, how do species arise? Please remember, everything that I have said so far tells you how you might get this sort of optimal engineering design. But how do you go from one optimum to a second radically different optimum? After all, if you believe in evolution, in this modification by descent, you know that at some stage there were creatures that looked like fish, and later creatures that looked like amphibians, and later creatures that looked like reptiles, and so on and so forth. Which were the descendants of today's fish and today's amphibians and today's reptiles? How did that happen? Darwin's answer, and one should say this is the answer given by most, well, people who think along the lines of the modern synthesis even today, is that essentially you had more of the same. Meaning, you had divergences, adaptive changes in different lineages, and these adaptive changes diverged if due to some reason or the other, members of these lineages were unable to interbreed. So for example, if there was some change in the pattern of distribution of water, let us say, so that two subpopulations of the same population came to be separated by a river on account of a flood and a new channel being opened up, these two subpopulations would be free to evolve in their own directions without their gene pools mixing. And perhaps the conditions in these two areas differed so that they achieved slightly different optima, and so on and so forth, until you got complete divergence. Right. So I end the first part of my talk there. What I've tried to present to you is the following. There's a coherent theory of evolution originally due to Darwin and Wallace, buttressed with a huge amount of study from essentially all areas of biology known to us today, which point to the following fact. There are things called genes. These genes, with the help of accessory proteins, can make copies of themselves. In addition, these genes can somehow catalyze the formation of proteins. So they make use of substrates. They work as enzymes, if you wish, in some sense, to help the synthesis of these proteins. Proteins build bodies. Bodies differ from each other, primarily because genes differ from each other. Genes differ from each other because of random causes, so-called mutations, as well as because of these shuffling variations caused by shuffling. Therefore, proteins differ from each other. Therefore, bodies differ from each other. Now comes the central assumption, which is that some of these differences in these bodies, which are heritable, remember, because they're based on genes eventually, some of these differences in bodies make some individuals more efficient at survival and or reproduction than other individuals. Therefore, in the long term, the representation of these forms, these traits, these phenotypes varies from generation to generation. Because of the correlation of phenotypes with genotypes, evolution drags genotypes along with it. Therefore, gene frequencies change in the course of evolution. So this is the picture of evolution by natural selection in terms of the modern synthesis, where of course there's a huge amount of underpinning of mechanisms from molecular biology, biochemistry, etc., etc. So I stop there. We can wait for 2 minutes and then I come to the second part. Yes, please.
As you have given the example of the moth there, the dark color and the very faint color, so there was proof that the environment also affects the evolutionary process. My question is that as we are modeling mathematical...
Be careful. The environment acts, if you wish, as a selective agent, and in that sense affects the evolutionary process. It's not a direct player in the change.
So this is not a direct player?
In this picture, no. But you will come to see something in a moment now where it may be a direct player.
Okay.
What I've said so far, in terms of the modern synthesis, it's very much not a direct player. It's a bystander.
Okay.
So not in this sense a passive bystander. You know what I mean?
Okay.
So if the eukaryotes and higher organisms evolved from bacteria, that is the view, right? I mean, bacteria are more primitive.
Oh, no, not really. So things get complicated. I didn't want to get into this at all. So the current picture we have is that if you, it impinges a little bit on what I was going to say. The current picture we have is that if you go back sufficiently far, which may be as much as 4 billion years ago, then you, A, didn't have these distinct lineages, and B, there was so much gene exchange between organisms that you couldn't even define separate lineages. So it's a melting pot if you go back sufficiently far. Please.
As you mentioned, there are some random effects that causes different genes, or causes some effects on genes. Where those random effects come from?
Various causes. The easiest to understand, I think, is chemistry. Everything that goes inside the cell depends on chemistry, depends on reactions in which molecules collide with other molecules. There's a binding energy involved. Because of thermodynamics, right? You can get different binding energies between different sets of molecules, right? Therefore, depending on what the difference in binding energies is, you can get the right binding or the wrong binding. Am I making myself clear? So that's one. So chemical accidents, that's one possible source of mutation. There are other sources of mutations which we will look at. Okay. Now, radioactivity is commonly known to be a cause of mutational change, so it can actually make holes in the genome. Right?
But the environment can be a very important thing here, because as you said, for example, radioactive comes from environment. So those random effects can cause when you look at many number of species in in the same environment, it has the effects is distributed normally in a Gaussian distribution.
No, please
as you
don't
know.
use words. It distributed in some way, let's say. Distributed in some way, okay.
The effect, for example, when we have a number of different, for example, butterflies in the same environment, most of them must be like each other.
You are actually touching on something different, which I was not planning to address in this class at all, which is the following. Also, I'll just pose it in the form of a question because I won't address it. Maybe we can talk about it later. The question is the following. If you look at genetic variation among organisms, butterflies of a kind, as you said, there's a huge extent of genetic variation, measurable today. If you look at variation in forms, it's hardly that much. It's very little. So the important question is, why is so much genetic variation masked? And it happens to be. I'll let you think over that as a question, but that is a big question, that is a fact, and we think we know at least some of the answers to the question.
Thank you.
Right. Oops. Yeah, so these alternatives to natural selection are many. The most important in terms of the amount of time that's been devoted to it, and in terms of the fact that a great deal of molecular change, by which I mean changes in DNA and proteins, not necessarily changes in forms, again comes to a part of what you said, a great deal of molecular change seems to be based on this concept of neutral molecular evolution. In fact, it essentially takes this idea of Sewell Wright a step further. Wright said that accidents, some individuals having many children, other individuals having no children, death by accident, etc., accidents can cause changes in gene frequencies which are not predictable, which are, as one says, stochastic. Moto Kimura said there's one type of accident which is particularly important, which is a change in the primary DNA composition which does not lead to any obvious change in the protein which results from that DNA. Now, this is something which we know immediately is possible, because in going from DNA to proteins, you go codon by codon. Different codons specify different amino acids, by what's called the genetic code. And we know that the genetic code is highly degenerate. Therefore, several codons can specify the same amino acid, which means that mutations which result in a codon changing to an equivalent codon, what's called a synonymous codon, will not have any change in amino acid, and therefore might not have any phenotypic effects, might not have any consequences at all, because you have the same primary amino acid sequence. Turns out that the real situation is somewhat more complex, but for the moment I'll let it be. Kimura went further and said maybe there were many other changes in which the DNA sequence changed, the amino acid changed, therefore the protein also changed, but it had no effect. And this kind of thing is called neutral evolution, and that cartoon is a rather nice cartoon. You can look at it later. It's Kimura's own. Neutral evolution leads to the consequence that you can now see there's a trade-off between the strength of natural selection, the rate at which mutations occur, and population size. And here one is speaking of the population either of organisms themselves that exchange genes, that reproduce with each other, or the population of genes, which is usually at any given genetic locus twice the number of organisms, simply because we have 2 copies of every gene. And what this picture tells you really, and I'll put it in words and let you figure it out slowly later, is that given 2 alternative genes at a locus, alleles as they are called, one of them being more advantageous than the other, in a sufficiently small population, and depending on the intensity of selection, either of 2 consequences can follow, 2 non-intuitive consequences can follow. Either the more advantageous allele can get lost, or the less advantageous allele can get fixed, as it said, which means spread through the entire population. And what this curve tells you is the way this happens. If you see the green line on top here, it tells you what happens in an infinitely large population when the situation is completely deterministic. These stochastic effects play no role whatsoever. But in finite populations, and what finite means in this case is defined precisely, it has to do with the product of the population size and the selective coefficient, or the product of the population size and the rate of mutation. So in finite populations, as defined here, it turns out that you can get this counterintuitive effect, which means that there are changes that might occur. Now, what changes? Whole variety of changes. I think Lynch is one of the speakers that is due to come and lecture in this school later, and he'll probably expand on this a great deal. One moment, please, I'll come to you. So the changes can be primary mutations of the kind that I told you. They can be things like parasitic DNA entering the genome, DNA from somewhere else, maybe a virus entering the genome. They can be things like non-coding DNA, which comes in simply because the sequence gets mutated by duplication, right? So there's a genetic change, and this duplicated DNA accumulates mutations now, becomes useless because there's a good functional gene around. So now you have a piece of non-coding DNA and all kinds of other things. And if you look at the structure of genomes now, you see how bizarre they are in the sense that they're not theoretically something that one would have predicted before one saw them. The existence of introns, the existence of, I've already said things that code for proteins, but the proteins don't amount to anything. Stretches of DNA that code for RNA, which does not code for protein, so the cell is doing something, but why? Stretches of DNA that code for RNA, which has a regulatory role, all kinds of complicated outcomes, which you can explain certainly on paper by saying that these are things that happened when populations were very small. They were deleterious, meaning that they lowered reproductive fitness, but it didn't matter precisely because the population was small, so they spread to fixation and now you're stuck with them. Of course, later you could get secondary genetic changes which ameliorated these deleterious effects, and so the situation begins anew. Yeah, please.
Those changes are not. It is not due to prevalence of those genes to undergo some specific biochemical reaction.
As I said, so this is the same question or the similar question to what was asked earlier. As far as we know, there's absolutely no tendency, right, in living organisms to change in a particular direction, as far as we know.
And what about the interactions?
What about?
What about physical interactions?
With great deal. That is certainly very important, yes. I'm going to come to that in a moment now. You'll see that. That is very important. Physical interactions between individuals, you mean? Yes. Yeah. Yeah, please. Quickly though. I'll finish and then we can discuss it.
Hello.
Well, in many ways. So one simple way would be to say, maybe there's a protein. Remember, proteins are folded up in complicated ways. Maybe there's a protein in which its surface is exposed to other proteins and other things in the cell. And the surface is all important in terms of whatever role that protein plays. So maybe if it's an enzyme, it's an active site consisting of half a dozen amino acids. The inside of the protein has to consist of hydrophobic amino acids simply to keep the proper folding structure. So if you change one hydrophobic amino acid inside for another hydrophobic amino acid, maybe it doesn't matter. For example. Right, so this really turns out to be, the mathematics of this is rather nice and the formula is straightforward. The population times size times the selection coefficient has to be less than 1. There's a factor of 4 for reasons that we don't need to get into. So the consequence of this is, as I said, you can get fixation of suboptimal alleles, you can get invasion, successful invasion by parasitic DNA, and you can get what I've called Baroque embellishments, the kinds of things that you will come across if you look at the molecular biology of the cell. Really pretty bizarre things, some of them, right? Now, what are the other consequences that follow from this sort of a neutral picture? There are a great many. Bahram Hoshmanzadeh has worked out many of them in detail, and he has shown, for example, that you can get patterns of clustering. So this has nothing to do with DNA. This actually has to do with real cells and the way they spread on a plate. And you can see the clusters there on top, which are most attractively explained by saying that the cells are moving towards each other, but in fact turns out in many cases to be explainable by a neutral model by in fact a random walk assumption. There are neutral interaction networks. In other words, what you can do, and this is the paper by Arpa and Erzan, what they did was to take a messenger RNA molecule, and for those of you who aren't familiar, now we know that messenger RNAs, which are the intermediates between DNA and protein, these messenger RNAs can be regulated in several ways by what are called small interfering RNAs. And the picture here is for a particular form of this small interfering RNA called microRNA. So the question now is, suppose you take a messenger RNA sequence, some sequence. So all of this is an experiment done on a computer. And now you ask yourself, given the kinds of things we were talking about a while earlier, binding energies, right? How many kinds of microRNAs of a certain size, a reasonable size so that your computer doesn't break up? So let's say 7 bases in length. How many kinds of microRNAs can you get that can interact with this arbitrary messenger RNA purely on statistical grounds and interact strongly enough to affect the functioning of this messenger RNA? You can do this experiment. It's a straightforward computer experiment. And that's the kind of probability distribution that you get, which you see there, which is one of the 2 black lines. There is some massaging of the results which explains the difference between the 2 black lines. But the point there is the red lines are the real data. So you see an impressive level of correspondence between the sorts of outcomes that you expect on a neutral assumption and the actual outcome that you get, at least in terms of this frequency distribution, in real organisms, which makes you wonder, of course, is it possible that you don't need to go look far beyond statistical probabilities in explaining why you get this form of microRNA regulation, this precise form of microRNA regulation for so many messenger RNA molecules? Something else which is really, historically, this is extremely important, a whole lot of studies done by a man called Leigh Van Walen, who was a paleontologist at the University of Chicago. And Van Walen extended the following finding. The finding which paleontologists had made much before then, and this is also something worth thinking of. I'll express the finding in 2 slightly different ways. One way to think of it is to say that most living species that have ever existed are extinct. Most once living species that have ever existed are extinct. Okay. You can make it more quantitatively. The more extreme paleontologists do so. They say, and I've heard different figures depending on who I read or listen to, they say up to 99% of all living species that have ever existed have gone extinct. Van Walen went a step further and he said, and remember there's a massive amount of fossil data available and was already, this work is about 40 years old now. Van Walen went ahead and said, okay, if these species have gone extinct, if this many species have gone extinct, and in fact he worked at different levels as well, orders, genera, larger groups, and so on. If they have gone extinct, they've had a certain tenure on Earth. So they've had a certain lifetime on Earth. By putting together these lifetimes and arranging everything in one picture, can I work out what the probability of extinction of an average species might be? And he's done that for a great many groups. I've given the reference there. His paper is even today worth reading. To sum it up, what he found was that the probability of extinction per unit time when plotted on a semi-log plot was a straight line. In other words, exactly like the kind of curve you would get in a picture of radioactive decay, which seemed to suggest that you could purely mathematically explain these patterns by saying that there was a constant probability per unit time, per unit species, if you wish, that the species would decay, that the species would go extinct. Now, this is a bit puzzling for the following reason, that intuitively you would think, and all 3 of the observations that I've made here are puzzling. Intuitively you might think that the longer a species has been around, the better it is at coping with whatever problems confront it in its environment. And therefore that these distributions ideally should have very, very long tails, very long, what they don't seem to have. And Van Walen's own verbal explanation for this has caught on in the literature, and the phrase is now used for a great many other things. He called this the law of the Red Queen from Alice in Wonderland. And he said, look, as species continue staying on Earth, environments also deteriorate. Deteriorate meaning in terms of whatever the species was adapted to initially in the beginning. And therefore, there's a certain kind of lack of resilience on the part of these species as times go on, because environments keep changing, right? And as a result of that, you end up with what looks like a radioactive decay law. This red queen hypothesis now is used in a great many contexts, basically to try and explain stasis, to explain why in many situations things don't seem to change. And the idea there is things might have changed were it not for changes in environments which reverse the effects of that change. Right. I think the culmination of these neutralist ideas of evolution really, and this is already, once again, more than 35 years ago, this particular picture, the culmination was the idea that you could build evolutionary trees by looking at similarities and differences, exactly of the kind that I mentioned earlier. But now, similarities and differences in DNA molecules, meaning in DNA sequences, or in protein sequences. This is for a protein known as cytochrome C, and this is famous as one of the first such evolutionary trees that was built. Now, the difference is the following. The conventional evolutionary tree that you built was based on similarities and differences and was later used by Darwin and Wallace and a great many others to say, aha, this can be explained by evolution on the basis of evolution by natural selection. This evolutionary tree, on the other hand, that you see is built on the assumption that you don't need natural selection, that what's happening is neutral evolution, so that these distances that you're measuring are random walk distances that have taken place, and yet the trees are very similar. The trees are very similar. So this is something for you to ponder on. I'm not going to spend time on this here. This has been remarked on a great many times and discussed a great many times. So the fact is that evolution seems to work at two levels. One is the level of DNA and proteins, where the overwhelming change is a change that does not result in changes in phenotypes. The second level at which evolution acts depends on changes in phenotypes, right? But the evolutionary trees that you get from these two pictures are the same. And as I said, I'm not going to expand on this here. John Bonner has gone the furthest in this direction. Remember, we're talking about neutralist models. He has gone the furthest in this direction by saying that at least among microorganisms, so these are these single-celled organisms that I talked about earlier, which were not bacteria. So these are eukaryotes. Bacteria, by the way, are still very difficult for evolutionary biologists to deal with. So most of the time, we prefer not to deal with them. Just look at them with respect and say, you stay in your world. Though they have become very useful in terms of experimental evolution studies, which is a different topic altogether. Anyway, John Bonner says that these nucleated single-cellular forms, which come in an amazing number of varieties within the same broad grouping. So these are what are called foraminifera, the ones you see in the picture there, from an ancient picture, from an ancient set of observations made by Haeckel. Bonner says that these forms, which look extraordinarily different from each other, and they reproduce, faithfully and so on and so forth, he claims that these forms may have been neutral as well in terms of their form. So what he's trying to say now is that in addition to a random walk in genetic space, which we've been talking about all along, perhaps simple organisms can perform random walks in phenotypic space as well. Right? Please try and ponder over this. So earlier I said genetic changes, protein changes, no changes in form. Bonner is saying, yeah, maybe there are changes in form as well, but they too could be random and yet get away. So that is an extreme thing. Right. Now I'm just going to rapidly go through a checklist, as it were, of other kinds of ideas which are creating excitement among evolutionary biologists. I can't claim that that's only now as of today, but over the past, certainly over the past couple of decades or thereabouts. And some of them have come up fairly recently. So this thing called epigenetic inheritance. Remember, the modern synthesis depends essentially on hereditary traits being ascribable to DNA, and therefore changes in hereditary traits being ascribable, at least to some extent, that's the idea of regression, remember, changes in hereditary traits being ascribable to some extent to changes in DNA. Epigenetic inheritance says that A, there could be changes in form in this case, not involving DNA changes, which is not too difficult to understand. You can think of all sorts of reasons why things might change, forms might change. But more interestingly, changes which are not due to DNA changes being heritable, so that you have a form of inheritance now which is not DNA based. And this is a classic experiment done by Sonneborn and Bison involving what's called ciliate, a complex single-celled organism which exhibits enormous details of structure on its surface. And these are fellows that swim in the water. In order to swim, they make use of what you might call oars, right? So these are cilia, which are synchronized to beat in the same direction. And these cilia are arranged along a whole number of vertical rows. You can see those rows there. They are the ones which light up as points in this picture. So all the cilia have the same polarity, therefore they beat in the same direction. Sonneborn and Bison cut out an entire ciliary row, inverted it by 180 degrees and saw what would happen. Now with ciliates you can do these experiments and get away with it, because it has a tough cortex and it heals naturally. So now you had a paramecium or a stentor or a euglena. They worked with many different ciliates. Now you had a ciliate which has, let us say, 9 ciliary rows in which the oars beat in the same way, and the 10th one in which they beat in the opposite way because you've inverted it, right? Now what happens? Well, of course, it isn't as good a swimmer as the others, but 1 in 10 isn't too serious either, so it can get by. But the interesting thing is when this fellow divides, and it's asexual division, so it's a faithful replication of itself. When it divides with this inversion still in place. So here you have a nice example of reproduction, which is faithful, which is not based on DNA. Now, great many other examples. I've chosen this one simply because it's of historical interest and it looks fascinating. There's a functional correlate immediately. So epigenetic inheritance is very, very common among human beings. The most famous recorded case now has to do with what's called the consequences of the Dutch famine, where a great many women who were in Holland, who were pregnant around the end of the Second World War, when the country was hit by famine, gave birth to babies who were severely underweight. No surprise in that. Terrible nutritional status of the mother, therefore underweight babies. But what was interesting was that these underweight babies, who later grew up, of course, and came of reproductive age in the 60s and 70s, also had underweight babies. And this study continued for 2 generations as far as I recollect. They had, so we went to a second grandchildren generation as well, also had underweight babies. And when I say underweight, I mean relative to the population average. So this is a nice example of phenotypic inheritance, or as I've said here, epigenetic inheritance in human beings. Many examples now. We know that it works. I just leave you with one idea. One thing that is not clear yet is whether this kind of inheritance can form the raw material for natural selection. And why do I say that? I say that because unlike mutational change, which is essentially permanent, once a change occurs, it's there forever. And by that I mean that the time scale of a mutational change, typically in units of generation time, which is the time that operates replication time, effective replication time across generations, the time scale of mutational change per gene is of the order of 10 to the 5, 10 to the 6, that kind of generation. So once a change occurs, it's there for the next 100,000 or million generations. Therefore, natural selection can do whatever it wants, knowing that it's got a stable gene to work with for that long. It's not clear that epigenetic changes have that high a lifetime. This still remains one sticking point. There are ways in which people get around this sticking point and say, nonetheless, they could have evolutionary implications. We might discuss that later if you like, but I won't go into it. Right. Someone was asking about big mutations. Now, this is something dramatic, chromosomal mutations. And this is an example done close to where we are in Mysore, where Ramchandra Rangnath and colleagues showed that the flies that they were able to collect from the wild, and I must say by they, I mean really a great many people, including some that happens in South America. The flies, fruit flies that they were able to collect from the wild turned out to fall into different categories when they looked at their chromosomal constitution. Those are the red and yellow lines that you see on the screen there. And it was very easy to see that one chromosomal constitution, please remember, looked like another chromosomal constitution, except that some of the chromosomes in one had fused to give rise to the chromosomes in the other one. So what you had here was a mutation. Sometimes these things are called macro mutations, major mutation, not a single base pair change, but a change in which 2 chromosomes in a parent gave rise to 1 chromosome in a progeny. This can happen due to what are called meiotic errors during mistakes made during replication. So there's no ascribable cause except to say that there's an accident. By the way, a very common thing which leads to fairly serious consequences in human beings due to something similar, not chromosomal fusion though, is called Down syndrome, where occasionally mothers have babies who have 3 copies of a particular chromosome instead of 2, chromosome number 21. And there are interesting, so there's an epigenetic effect here as well, in the sense that mothers beyond a certain age are prone to this sort of error, which is called a meiotic disjunction. Here, this is a meiotic fusion that we saw. Anyway, for our story, what's interesting is that once this happens, these 2 individuals cannot successfully breed with each other. And the reason they cannot do that is because if there's a mating between these 2 individuals with these different chromosomal constitutions, the chromosomes cannot pair correctly and therefore cannot give rise to the right kind of germ cells, which is the egg cells and the sperm cells. So basically the hybrid here either does not form at all, you know, if the mistakes are huge, or if the differences are huge, or forms but is sterile. So it's a no-go situation. Interestingly, you know, this plays the role of what I mentioned earlier as a river coming in and separating 2 subpopulations, because now you have 2 subpopulations, one with the original chromosomal constitution and the other with this new changed chromosomal constitution. And these can evolve differently. And in fact, it looks as if these 2 fellows are behaving like incipient species. This is where you get into problems of nomenclature and when do you really call 2 things different species, etc., which I said I didn't want to get into. But these certainly look like incipient species. And in fact, you can test that by carrying out crosses between members of these pairs. And I must say here that there are many different pairs that are used, not just the first 2 that I talked about. These things have come to be called races in the biological literature. And because these are races based on chromosomal differences, cytological differences, they are called cytoraces. So these are cytoraces of Drosophila. And you can see immediately they have isolation indices, meaning problems with mating and giving rise to fertile young, which can vary from very small to very high. Certainly all of them are reasonably high. So there are reproductive barriers that come out. By the way, what you need to compare here is the percentage of so-called homogamic matings that are successful with the percentage of heterogametic matings. And you see immediately the differences that are there. So that's the second difference. Third kind of thing, and this is simply a cartoon, this does not give you the full message. Third kind of thing is the following. And this is where you need to look at the color coding. There are a great many organisms shown on the right-hand side. Turns out that time and time again, you find the same genes showing up in different organisms. No surprise, because we said evolution is modification by descent. But these same genes sometimes do different things in different organisms, meaning that the proteins that are coded by these genes subserve different functions. And more often, these same genes turn out to be regulated in different ways so that different subsets of them, so the same genes, please remember, meaning the same protein coding genes, different subsets of them function in different pathways in different organisms. So now you've got a new kind of repertoire available to evolution over and above what was available via mutation. And this is via combinatorics. So you can use the same genes, same protein coding product, but fool around with them in different ways. And as a twist to this story, if you come to the bottom of the picture, and there you see a mouse and a kind of fish that is very popular in molecular biology studies today called the zebrafish. You see that not only do you have the same genes in them as you had, for example, in the lancelet, which is somewhere on top, or in the worm, roundworm, which is on top. Not only do you have the same gene, but you have the same gene in multiple copies. So these are features of evolution that are not easily explained by natural selection. So what I'm trying to tell you here is that there is more to the genetic repertoire available for evolution to take place than point mutations in single genes and simple associations between genes that the modern synthesis depended on. This is something which has become notorious now. Heredity itself has been called into question because heredity simply meant there were parents, offspring, they were parents, offspring, they were parents, their offspring, etc., etc. Now it turns out that certainly in unicellular organisms, many unicellular organisms, overwhelmingly so in bacteria, there's sufficient evidence that genes have moved horizontally, which is to say not necessarily between parents and offspring, but between any two individuals in a given generation. And more to the point, even across species, even across large bacterial groups, this is known to have occurred. Now, clearly, this means that the picture of evolution by natural selection as we posed it cannot be correct. In particular, that tree of life can no longer be a tree to the extent that this is true. It becomes a net, you can see immediately, because there are horizontal lines connecting different branches. In a sense, you might say, fortunately for us, horizontal gene transfer seems to be pretty rare, if at all it exists among us, among eukaryotes in general. Us, meaning everything from the yeast to us. Right. Something else. We talked about mutation as a source of variation, recombination as a source of variation. It turns out that gene expression itself is a source of variation because it's not determinate. Meaning, a gene might code for a certain protein. But all that you can say with confidence is that that gene, that DNA sequence, is competent or capable of coding for that protein. Turns out, you cannot bet that it will code for that protein every time it could, nor that it will code for that protein in every cell, in every place where it could. So this picture gives you an example of the same gene in different cells, and these cells are what are called clones, so they're genetically identical. Same gene in a clone of cells, same environment, but in some cells the gene is on, some cells the gene is off. So the color there is due to tricks that you can use now to see whether the protein product is being made or not. And in fact, it turns out that these are temporal fluctuations. So the fellow who is bright yellow there can go dark the next moment. One of the dark ones can become yellow. So you get an off, on, off kind of pattern. Gene expression is stochastic. Now, clearly this means that to the extent that gene expression translates into phenotype, and if that translation occurs within a time shorter than the time of switching of gene expression, you've got phenotypic variation. Therefore, you can get selection acting on it. Can that lead to evolution? That's still an open question. But clearly you can get selection acting on it. And indeed it does. There's a nice example from a paper of Hardicker's where the same kind of thing, the colors mean the same thing that they did last time, except that the red and green colors here have functional consequences. And it turns out that the ones that are colored red make a receptor. The ones that are colored green make a molecule which binds to the receptor. And the fellows that make the molecule move towards the fellow that makes the receptor. From a collection of single cells, which were genetically identical, in the same environment, you get heterogeneity appearing, and this heterogeneity has a functional consequence because you can build a tissue now. All of these fellows come together and form a tissue. So you see there are interesting consequences of change here, which certainly don't have anything to do with genetic change in the way that I talked about. Now someone asked about the environment directly affecting something. It turns out that does too, that happens too. Again, many cases of this known. This is a popular case from the literature, from a plant known as the lake cress. So it's a single plant. The genetic variations have been seen. And the leaves of this plant, which happen to be underwater, look completely different from the leaves that are above water. And you can see these shape changes. These are very clearly epigenetic effects due to differential gene expression of the same sets of genes, but these are not heritable, meaning that you cannot now claim to say that you will breed true from either of them. Nonetheless, this shows variation, and it shows that there is a certain amount of repertoire available even within a constant genetic makeup. This is the point that phenotypic repertoire available. This is a dramatic example which I like a lot, discovered by Lakshmi Namdar. It's well known that sex, which of course is an extraordinarily important phenotype for creatures like us, it's well known that sex in some organisms does not have a genetic basis in the usual sense of the term. Rather, it has a chromosomal basis. In yet other organisms, it has neither a genetic basis nor a chromosomal basis. It has an environmental basis. And these environmental forms of sex development of sex are enormous. They are a fascinating topic in themselves. I could tell you some more later if you want. The one of interest to us, which is found in many crocodilians and tortoises, turtles, is temperature-dependent sex. And this too comes in different varieties. You have sharp threshold temperatures. Eggs incubated above a threshold give rise to males. Eggs below that temperature give rise to females. Right? In other cases, eggs above the temperature females, eggs below the temperature males. Sharp thresholds, again remember, these are, the experiments can be reproduced in the lab, of course. What Lakshmi Namdar found is that you have these, please look at, follow them with me, let's look at the red curve. 1, 2 thresholds in going from male to female and 2 thresholds in going from female to male. Meaning, at a low temperature you get one sex, cross the threshold, other sex, cross a second threshold, you come to the first sex. You get my point? And it can work the other way around. So, enormous phenotypic variation, no genetic variation, clear evolutionary consequences here, because if you have progeny of a single sex, then that line disappears. So you need to pay attention there. This, of course, this picture I've put just for the sake of its dramatic effect. Co-evolution is now being looked at seriously, which means our earlier picture had an organism, the environment throws up certain challenges, and that organism, in the sense that we described it, responds to those challenges, meaning its lineage responds to those challenges, not the organism itself. Now it turns out that among the challenges that organisms face, and face very frequently, are other organisms. Someone raised this as a question a while earlier. Now those other organisms can belong to its own species, they can be its cohorts, or they can belong to other species, right? And the examples now piling up of groups of organisms evolving together in this fashion is enormous. Huge number of examples. So what does this mean? From the point of view of the first organism, the second organism is its environment, and vice versa. So here you have a situation in which the environment is no longer fixed. The environment also keeps varying. So what you have is co-evolution between organism and environment, and clearly that's a slightly trickier question than that of a single organism evolving. By the way, this picture too had been studied in great detail some 50 years ago now, 60 years ago now, when people noticed that parasites tended to co-evolve with their hosts. So we are co-evolving with the parasites within us. And now, thanks to the techniques of molecular biology, this particular line of study has become very popular in evolutionary biology with regard to human health and disease, especially infectious disease, and the co-evolution of human beings and disease-causing agents is a popular example of it. This, as I said, I put that as a dramatic example. How does the bird know that the crocodile is not going to shut its mouth? Well, it knows that because this is evolved as a behavioral trait, and interestingly enough, it works only in particular places. So the crocodile has so-called cleaning stations, where the dentist shows up and cleans its teeth. If the dentist goes somewhere else, it's finished. And in these cleaning stations, it turns out there are certain times. So you have to go at the right time of the day, in the right place, the cleaning works, the bird flies off with whatever it's managed to get from the mouth of the crocodile. The crocodile is happy, it doesn't get teeth decay, no root canal jobs for it, and so on. So I'm going to stop there. Let me finish. I had a couple of other alternatives to natural selection, but you can look them up later. Let me try and tell you what it is that I've really tried to convey in this second part, which is evolution by natural selection remains the default option, as I said, for most evolutionary biologists. Interestingly, this is a personal observation, most biologists who do not do evolutionary biology seem to be convinced that evolution by natural selection is the only explanation for evolution. Among evolutionary biologists, there are graded differences of opinion between those who think that natural selection, even today, has not been supplanted by a sufficiently interesting alternative, on the one hand, and on the other hand, by people who think you really have to include natural selection as just one element among a whole host of alternatives, some of which I've illustrated here, which are needed to properly understand evolution. And there are different phrases used to describe these alternatives. One popular one in the literature these days is extended evolutionary synthesis. So the idea being you bring in epigenetics, you bring in horizontal gene transfer, you bring in developmental effects of the kind that I showed and so on. Put all of that in. Strict selectionists would still say that these are at the fringes of the core of the theory. It still holds good. Two further points before I end. One point, there's a critical difference, it seems to me, in 2 or 3 ways in which natural selection and the modern synthesis look at evolution, and in the ways these alternatives say you must look at them. So let me just highlight these points. One point is the role of the environment, which you can now use within quotation marks, role of the environment in classical theory versus the role of the environment in one of these many alternative theories. So now this is an interactionist environment. It's an activist environment, a middlesome environment. There it was an environment which just sifted and chose. In other words, the classical picture of natural selection is an externalist picture. There's an environment and it's up to you to respond to that environment. The current picture is more of an internalist picture with, of course, also an externalist element to it. So internal changes fuel a lot of evolutionary change. So that's one aspect. Secondly, natural selection ipso facto, and the hill climbing, mountain climbing metaphor is useful here. Natural selection ipso facto has to be a slow process. The idea is very simple. If you are on a local peak, or anywhere for that matter, and close your eyes and take a big jump, the chances of your landing on a peak are very, very little. Right? So natural selection has to be slow, has to be gradual. And in fact, if there's one thing that Darwin emphasized again and again and again in his book, it was the slowness of the process of evolution. He said, if there's one thing that you could show me which would disprove my theory, which would make me give up my theory, that would be that selection has acted rapidly. On the other hand, these alternative theories make no such demands at all, as you can see. They do allow for rapid changes. Right? Second point. Third point, very interesting. The classical theory of natural selection says genetic changes occur first, randomly. Some of those changes are advantageous. The advantageous ones spread through populations, and gradually they are built upon, one upon the second, upon the third, upon the fourth, and so on and so forth, so that you get cumulative changes finally, which you and I can recognize, these are real big variations. We can see these differences. So genetic change, accumulation of advantageous changes, I should say accumulation of consequences of advantageous changes, large-scale variation, macroscopic variation. In any case, genetic change precedes variation. As you will have seen in many of the pictures that I've presented here, these alternative mechanisms are also open to the idea of phenotypic change, form change, behavior change, especially behavior change, preceding genetic change. Because the same genotype is consistent with so many phenotypes. Right? Now, if that can happen, you have a rather nice route for natural selection to act subsequently. Because natural selection can act now not so much as the vehicle of phenotypic change, but rather as something which stabilizes an advantageous phenotypic change once it has occurred. So it becomes more of a stabilizing agent primarily than as an agent for change, which is an interesting thought. And as I showed many slides ago, unfortunately I rushed through them, these experiments of natural selection under natural conditions in the wild show that stabilizing selection is really quite often seen. So perhaps, this is imaginative now, perhaps changes, or many changes might have occurred in that way, occurred initially by virtue of physical reasons, by virtue of stochastic reasons, by virtue of phenotypic noise, what have you, and later been buttressed by natural selection, which made those changes reliable. Please remember, all said and done, genetic changes are reliable in a way environmental induced changes are not, because the environmental can change in a moment, whereas genetic changes are stable at least over that time scale. So keep these two differences in mind. One last sentence, evolutionary thinking in the broad sense now has permeated a great many areas outside biology. So evolutionary psychology, of course, you keep reading about this in the newspapers as well. Historians talk about evolutionary theories. By that I mean they're really thinking of selectionist theories vis-a-vis stochastic theories, right? Cultural evolution is a popular, very intense study now, because anthropologists and sociologists have now started seriously looking at the possibility, which is there of course, that cultural evolution, which all along was believed to be delinked from biological evolution, is now seen to depend on biology as an essential substrate. And there are really quite dramatic examples of this. So for example, the culture of rearing cattle, of dairy farming. You can see immediately that you have two hypotheses. People keep cattle because they drank milk. You know, they wanted milk. But the other hypothesis is they drink milk because they keep cattle, which seems a bit strange, but that too can be followed up seriously. So cultural evolutionists have started to look at evolution intensely. So I end with that, and I wish you happy journeying at least over the next 2 weeks through evolution land. Thank you.
Hi, thank you very much. Can you just touch or give some expertise on this aspect of synonymous changes? Like, we have always focused on largely on the non-synonymous changes, and we know that the amino acid substitutions are there, and even in human health and disease, but much less is known about the synonymous changes. Sure. I just wanted expertise on the synonymous changes. Like, currently we are finding, even as we sift through human data, that there are substitutions, synonymous substitutions that may be sort of causative variants for diseases, but not because much is not known, so it's just avoided.
Yeah, I don't want to go into this in great detail, but let me just mention the following. On the face of it, a synonymous change, so a change in a DNA sequence, which does not result in a change in a protein sequence or in an amino acid, a synonymous change ought to have no effect. But unfortunately, that's not necessarily the case, even on the basis of what we know of molecular biology. And the reason is the following. To go from a DNA change to a protein change, or to go from a DNA to a protein, requires many, many steps. One of those steps, for example, is to go from DNA to RNA. The other step is to go from RNA to protein. And let me focus on the RNA to protein step here, because it's the most relevant for this question. To go from RNA to protein, you need an entire accessory bag of tricks, also made up of RNA molecules. These are called transfer RNA molecules. Sometimes these are called literally the dictionary. So they are able to read words, if you wish, in RNA language and tell you or tell the cell what the equivalent word is in protein language. Now these dictionaries, because they recognize words in the RNA language alone, primarily, they can make distinctions between a codon and a synonymous codon. In other words, what you call a synonymous codon is no longer synonymous. Am I making myself clear? And one very easy way in which this can happen is the following. There may be a codon for which you have an entry in the dictionary. So you have an appropriate DNA molecule which reads that codon and says this is the amino acid. There may be a synonymous codon for which that dictionary element is missing. That tRNA simply doesn't exist, or exists but in fewer copy numbers, in which case the reading is less than efficient. So this is the most commonly known way in which synonymous codons can actually have phenotypic effects. There are other ways as well, but I won't go into them. I think the point is made.
I had a question regarding the examples you talked about in terms of epigenetic inheritance. Especially there was that interesting example of underweight babies across generations.
I didn't say epigenetic inheritance. I gave that as an example of natural selection working on humans.
I see. Oh, I thought it was a case of epigenetic inheritance. So.
Oh, I'm sorry. You're referring to the Dutch famine?
Yes.
Yes. True. I'm sorry. I misunderstood.
Yeah.
Yes, so, you're right.
Right. So in that case, what surprised me is here we are talking about a change in in a way, a change in form, in the sense that the baby is born at a lower weight than its peers. And if genes are not playing a role, then what is the basis for this inheritance?
See, I can't give you a detailed answer for this case, but in general, the following things are possible. Among other things that are affected in a baby, which is affected due to its mother's nutritional deprivation in pregnancy, among the things that are affected in the baby may be fertility-related factors. Okay? which are necessary, fertility-related factors, not reproduction per se, fertility-related factors, which are necessary for what are called the nurse cells in that baby. Now we are talking about a female baby, obviously. So fertility-related factors which act on the nurse cells in that baby, when the oocyte in that baby develops, so that oocyte comes up poorly endowed. You get the point? No.
Bifurcation happen in the tree? Like, I mean, when do you say that there's a bifurcation in the tree?
This comes to the tricky question of what you mean by a species. So today, most people, there are many definitions of what constitute different species or what the defining characteristics are. But most people would say one defining characteristic is the inability to mate with each other, inability to exchange genes, if you wish. Remember, I used the phrase gene pool. So these are two different gene pools. Now, why are there two different gene pools if there were one gene pool in the beginning? Right? And the answers to that can be many. I gave you one answer here in terms of gross chromosomal mutations, which simply make it impossible for the bearer of that mutation to mate with someone who doesn't carry that mutation straight away. But the bearer of the mutation is fertile in itself, and this one is fertile in itself. The second kind of answer, which is the one that many evolutionary biologists favor, though it doesn't have this kind of a dramatic demonstration, is geographical isolation, physical isolation, which simply makes it difficult for different individuals to come together to mate, and over the course of time makes them drift, if you wish, in the more colloquial sense, diverge in different directions. There's a third kind of answer which has become very popular in recent years. Since you raise it, let me mention this, which also brings in population size. This third answer is the following. Suppose you have a large population with a single gene pool. Suppose a small subpopulation from this large population is cut off due to whatever reason, again, some natural accident. This cut-off population being small in size will perforce have to go through cycles of inbreeding. There aren't too many individuals to mate with. Now, inbreeding is known to be a potent form of genetic stress, which means the products of inbreeding are far more variable than the products of outbreeding or outcrossing. Now, why is an interesting question in itself, which I won't even get into. But now we have a great deal of phenotypic variation available. You follow what I'm trying to say? Thanks to inbreeding. And there, if a particular variant, so there's not a mutation. So from that phenotypic variation, if one happens to be better adapted than others, even if it's very different from the parental population, it can get stabilized in the course of time. So there are many of these possibilities have been discussed.
Sir, you showed two pictures, different pictures, and told us the evolutionary tree are almost similar in both the pictures.
Molecular tree and standard conventional tree, yes.
Exactly.
Yes.
So this is just observation, or is there any interpretation
It's
or
an observation. It's an observation. Now the interpretation simply is, as you can think, let's suppose that there is a real, these are called phylogenetic trees as well. Suppose there's a real phylogenetic tree, which is the old-fashioned evolutionary tree. So what has been happening as this sort of evolution has taken place is that in parallel, but without any consequences for shape and form, in parallel, DNA sequences have been changing, protein sequences have been changing in ways that do not impinge on the form, but those changes have been occurring. You understand? So they can be used as signatures of this change.
Okay, and
Do you get what I'm saying? They don't reflect this change, but they have been occurring at the same time. So there's an internal change going on in creatures during the course of evolution as seen by you, and that internal change can be deciphered. Of course, you can't go back and decipher the change as it occurred. All of these are retrospective. These internal changes can be inferred from looking at DNA sequences and protein sequences. But they have taken place too, except that they're not reflected in the form directly. They're cryptic changes.
Thank you. The horizontal gene transfer, the tree of life cannot capture the events of horizontal gene.
Not the way that I showed it, no.
Huh?
No.
They can't capture the...
No, I mean, you can draw a structure with horizontal gene transfer, but it's topologically no longer a tree. It becomes a net, right? You have cross branches, so you have two branches and a cross branch between them.
What are the available tools to analyze genetic differences in order to capture the events of genetic gene transfer?
Well, what people do is the following. So I'll give you a simple example which is done. You can take the human genome, you can sequence the human genome, right? You can look at these gene sequences, you can do the same for a great many other organisms. You can look for resemblances, and you find, for example, this is a very popularly quoted example. You can find, for example, that within the human genome, you have a huge number of copies, in fact, many millions of copies of a short DNA sequence. So same DNA sequence, repeat, many millions of times, of a gene, of a DNA sequence, which is found in a virus, right? Not found in other mammals, for example. So the immediate hypothesis that springs to mind is that this must have entered via the virus. Now the second step is, how could it have done so? Fortunately for us, we know that there are viruses which can infect animals or plants, in fact, better studied in plants, but let's say in us, which carry as their active genetic material RNA, not DNA. Their genome is made of RNA. Now, these viruses have DNA intermediates, so in going from RNA to RNA, they need to make a DNA and then make an RNA copy. That DNA can integrate into the genome. These are called retroviruses. You must have maybe heard or read about them. They're especially popular now in the context of great many infectious diseases. So these retroviral genomes have mechanistically known routes of integrating into your genome. So the obvious inference is, when you find that viral sequence in your genome, you say it must have come from a virus. But now it's part of you.
Thank you. Great lecture. It seems to me that there is a gap in between the phenotypic and the genotypic when you talk about the evolution via changes in gene regulation, right? So you say we have the same gene, and then in different species, whatever that means, you have different things. Could this mean that there is another structure, let's say greater than a gene, which is playing a role here, or is there a way to start thinking about phenotypic in a systematic way that could allow us to do some progress in there? What's going on there?
Yeah, the first question, I'm not sure if it's meaningful to speak of another structure, I don't know. The second question, very much yes. So what you can do now is to look at the way in which genes are switched on and off, and you make a number of interesting discoveries. First of all, that genes do go through these changes. I'm not speaking of the stochastic changes, but I'm thinking of changes in which a gene is on for a very long time, or off for a very long time. Secondly, you discover that there are concordances and discordances. So when a gene is on for a very long time, you find that genes number 3, 7, 11, 18, and 26 are also on for a very long time. Thirdly, you discover that if you look at all these genes that are on for a very long time, you look at their DNA sequences, there seems to be something very similar in their DNA sequences. Not that part of it that codes for a protein, but that part of it which is close to the protein coding part and which regulates the protein coding part. So now you say, look, all of these regulatory DNA sequences are the same. Therefore, one regulator can simultaneously work on all of them. So you can see the scope for combinatorics here. Or alternatively, one regulator can act as a positive regulator, as an activator of this gene, and simultaneously as a repressor of that gene. So you have negative concordances, you have discordances. Those kinds of things are coming up. And this is an extremely vibrant area of study now among developmental biologists. Then of course, over and above that, there's this whole question of which genes are networked with which other genes, which proteins are networked with other proteins, similarly, along similar lines.